Imagine a world where every living thing begins with a single cell, a tiny universe, bustling with activity, preparing for the ultimate transformation. This is the story of life itself, the incredible journey of cell division. A process so precise, so powerful, it shapes every living being on Earth. But how does one cell become two? Or even four? What secrets lie within the nucleus, and how does life ensure its continuity? Buckle up, because today, we're diving deep into the microscopic world of mitosis and meiosis, the two epic tales of cell division that keep life moving forward, from the preparatory phase of interphase to the dramatic stages of prophase, metaphase, anaphase, and telophase. We'll unravel the mysteries of how cells divide, multiply, and create life as we know it. Ready to explore the science that powers existence? Let's begin. Cell division is the process by which a cell divides into two or more daughter cells. There are two main types of cell division. 1. Mitosis. A type of cell division in which parent cell divides into two daughter cells and number of chromosomes remain same. 2. Meiosis. A type of cell division in which parent cell divides into four daughter cells and number of chromosomes reduced to half. Before cell division, each cell undergoes by the process of preparatory phase is called interphase. Interphase. The cell grows, replicates its DNA, and prepares for cell division. Phases of mitosis. There are two main phases: karyokinesis and cytokinesis. 1. Karyokinesis. Division of nucleus is called karyokinesis. It is further divided into four phases: Prophase, Metaphase, Anaphase and Telophase. 1. Prophase. The chromatin condenses, and the nuclear envelope breaks down. Spindle fibers form. 2. Metaphase. The chromosomes line up at the center of the cell. 3. Anaphase. The sister chromatids separate and move to opposite poles. 4. Telophase. The nuclear envelope reforms, and the chromosomes uncoil. 2. Cytokinesis. The cytoplasm divides, and the cell splits into two daughter cells. Phases of meiosis. Meiosis complete in two cycles: meiosis I and meiosis II. 1. Meiosis I. 1. Prophase I. Homologous chromosomes pair up and exchange genetic material. The chromatin condenses, and the nuclear envelope breaks down. Spindle fibers form. 2. Metaphase I. Homologous chromosomes line up at the center of the cell. 3. Anaphase I. Homologous chromosomes separate and move to opposite poles. 4. Telophase I. Nuclear envelope reforms. Chromosomes uncoil to form chromatin. 2. Meiosis II. It is just like mitosis. 1. Prophase II. Nuclear envelope breaks down. 2. Metaphase II. Sister chromatids line up at the center of the cell. 3. Anaphase II. Sister chromatids separate and move to opposite poles. 4. Telophase II. Nuclear envelope reforms. 3. Cytokinesis. Cytoplasm divides, and the cell splits into four daughter cells. Thank you for joining us on this journey into the heart of biology. If you enjoyed exploring the wonders of cell division, don't forget to like, share, and subscribe for more fascinating insights into the science of life. Hit that notification bell so you never miss out on our latest video. Until next time, stay curious and keep discovering the magic of the microscopic world.